Welcome to the presentation of our online course, Digital Scale-Up and Optimization of Microbial Fermentations. Through our online courses series, you learn the principles of bioprocess engineering and their applications to enzymatic bioconversions, microbial fermentations, and animal cell cultures processes. For more details, visit us at bioprocessengineeringonline.com. The design and scale-up of microbial fermentation processes for the production of bio-based products across a wide range of industrial applications form the foundation of bioprocess engineering. The primary objective is to maximize the efficiency of microbial cells utilization within fermenters, with a particular focus on minimizing production costs while maintaining product quality and process robustness. A well-designed fermenter must be specifically tailored to the microbial biotransformation of interest, with careful consideration of the organism's kinetic and physiological characteristics. Key factors in fermenter design include selection of the appropriate operating mode, batch, fed batch, or continuous, depending on the process requirements and microbial cells' kinetics, optimization of operational parameters, such as substrate concentrations, feeding flow rate, induction timing for metabolite or protein expression, and fermentation duration. These factors play a decisive role in enhancing process efficiency, ensuring robust cell performance, and achieving optimal fermentation outcomes. Bioprocess engineering has evolved as a scientific discipline that provides a structured framework of knowledge, principles, and methodologies for the simulation, scale-up, and optimization of microbial fermentation processes. To accelerate process development and scale-up, bioprocess engineering employs mechanistic, knowledge-driven simulation models grounded in the understanding of key physicochemical and biological phenomena. These models serve as digital twins, virtual representations of fermenters, that enable in silico experimentation under various operating conditions. Typically developed from laboratory-scale experimental data, simulation models play a pivotal role in advancing scientific insight into microbial transformations. Furthermore, they play a critical role in the optimization of operating modes and process parameters, supporting the achievement of maximal productivity or minimal production costs. By enrolling in this course, you will develop a solid foundation in the digital methodologies of bioprocess engineering, with a focus on the scale-up and optimization of microbial fermentation processes. The course is structured in two key phases. One, model development from laboratory data. You will learn how to apply core bioprocess engineering principles to develop mechanistic simulation models based on laboratory-scale experiments. These models will be tailored to the specific microbial strain used, enabling you to capture and describe the key phenomena influencing microbial growth, substrate utilization, and product formation. 2, simulation-guided scale-up and optimization. Building on your model, you will explore how to use simulation tools to scale up fermentation processes and optimize operational strategies. Emphasis will be placed on selecting suitable fermenter technologies and operating modes, batch, fed batch, or continuous, to enhance the efficiency of microbial biocatalysts and maximize overall process productivity. Throughout the course, the principles and methodology of bioprocess engineering are illustrated through an industrial case study, the scale-up of Baker's yeast fermentation by Saccharomyces cerevisiae, from laboratory scale to a 100 cubic meter industrial fermenter. This case study provides a practical framework for understanding the analysis, simulation, scale-up, and digital optimization of fermentation processes, demonstrating how mechanistic models can be applied to support process development and industrial operation. The course is structured into two parts, comprising a total of 8 sessions. Part 1, Understanding and Simulating Fermentations. This section introduces the foundational principles and tools required to analyze and simulate microbial fermentations, Principles of fermentation engineering, kinetic analysis of fermentation, oxygen transfer and consumption in fermenters, construction of simulation model on spreadsheet. Part 2, scaling up and optimizing fermentations. This section focuses on applying simulation models to improve fermentation performance across different operational modes. Scale up and optimization of batch fermentations, optimization of continuous fermentations, optimization strategies for fed batch fermentations, sensitivity and uncertainty analysis of digital scale-up. The 8 sessions can be taken independently. In the first part of the course, you will gain a solid understanding of bioprocess engineering principles, concepts and methods relevant to microbial fermentations. You will explore the key phenomena that affect fermentation processes and advance in developing the fermentation simulation model based on fundamental knowledge. Session 1, specifically designed for newcomers to the field, provides a comprehensive introduction to the fundamental principles of bioprocess engineering. During this session, you will explore the following key topics: biochemical and physical phenomena governing microbial fermentations, mass balances in bioreactors, mass transfer phenomena in fermenters. In the second session, you will delve deeper into the kinetics of microbial fermentations with a focus on performing kinetic analysis in both batch and continuous fermenters, analyzing experimental data to determine fermentation rates for cell growth, substrate uptake, and metabolite production, calculating specific cellular rates to gain insights into microbial efficiency within the fermenter, understanding how these rates change over time. In the second session, you will also identify the kinetic laws and parameters governing the microbial cells used. These laws dictate the specific rates of cell growth, metabolite production, and substrate uptake, all of which are influenced by the composition of the medium, particularly the concentration of substrates. In Session 3, you will explore the continuous aeration process in aerobic fermentations. You will analyze the kinetics of oxygen transfer, solubilization, and consumption within the fermenter, as these are critical factors that often limit fermentation performance. You will also identify the rate law for oxygen consumption by cells, the thermodynamic law governing oxygen solubility in the medium, and the kinetic law of oxygen transfer from the air phase to the liquid medium. In Session 4, you will learn how to construct a simulation model for fermentation by combining the thermodynamic and kinetic laws identified earlier, along with the mass balance equations applicable to the fermenter's operating conditions. Additionally, you will learn how to numerically integrate the differential and algebraic equations that govern the time-dependent changes in biomass, substrate, and metabolite concentrations using a spreadsheet. The second part of the course focuses on the digital scale-up and optimization of the microbial fermentation. Using simulation models, you will analyze and compare the performance of the 3 principal modes of operation: batch, continuous, and fed batch fermentation. For each mode, you will investigate the influence of key operating variables and determine the optimal conditions that maximize 2 major performance criteria: fermenter productivity and sugar substrate to product conversion yield. In aerobic fermentations, achieving maximum productivity or process intensity requires careful consideration of oxygen transfer limitations. The attainable cell concentration is constrained by the oxygen transfer rate. The highest cell density is reached when the rate of oxygen consumption in the medium equals the maximum rate of oxygen transfer from the gas phase. That is, when the dissolved oxygen concentration drops to its minimum allowable level. You will learn how to determine this maximum cell concentration limited by oxygen transfer in all 3 operational modes: batch, continuous, and fed batch. Session 5 focuses on the digital scale-up and optimization of batch fermentation. Using the scaled-up simulation model, you will evaluate how key operating variables influence fermentation kinetics, oxygen availability, and the maximum biomass concentration achievable under oxygen transfer limitations. These variables include fermenter agitation speed, aeration rate, and the initial concentrations of substrate and biomass. Finally, you will determine the optimal operating conditions that maximize fermenter productivity and sugar to biomass conversion yield, while minimizing production cost. Session 6 focuses on the optimization of continuous fermentation, in which fresh nutrient medium is continuously supplied to the fermenter, while culture broth is simultaneously withdrawn. Using the scaled-up simulation model, you will investigate how the two key operating variables, medium feed flow rate and feed sugar concentration, influence both the transient behavior and steady-state performance of continuous fermentations. Through virtual experimentation, you will identify the optimal operating conditions that maximize continuous fermenter productivity and substrate to biomass conversion yield, while minimizing production costs. Session 7 focuses on the digital scale-up and optimization of fed-batch fermentation. In fed-batch processes, the fermenter is initially only partially filled, and fresh nutrient medium is progressively added throughout the fermentation. This operating mode allows the concentration of the limiting sugar substrate to be maintained at a low level, thereby reducing the formation of undesirable metabolites and improving process performance. Using the scaled-up simulation model, you will investigate how the three key operating variables, initial medium volume, feed substrate concentration, and target steady-state substrate concentration, influence both the transient fermentation kinetics and the final performance of the fed-batch process. Through virtual experimentation, you will identify the optimal operating conditions that maximize fermenter productivity and sugar to biomass conversion yield. In Session 8, you will learn how to perform sensitivity and uncertainty analyses within the framework of the digital scale-up of fermentation processes. The sensitivity analysis component investigates how variations in input data, such as microbial, kinetic parameters, and process operating variables, influence simulated fermentation kinetics and overall process performance. By quantifying and ranking the effects of these inputs, you will identify the parameters and variables that have the greatest impact on fermentation outcomes. This analysis highlights the factors that are most critical to monitor and control in order to achieve consistent process performance and reliable scale-up. In the second part of the session, you will apply uncertainty propagation methods to quantify the uncertainties or confidence intervals associated with simulated fermentation kinetics and predicted process performances. These uncertainties are determined from the estimated uncertainties in model parameters, operating variables, and other input data. By quantifying and ranking the contribution of each source of uncertainty, you will identify the variables and parameters that have the greatest impact on prediction accuracy. The online course resources include a series of recorded slideshow presentations that provide a comprehensive introduction to the principles and methods of digital scale-up for microbial fermentations. The presentations cover the following topics: influencing phenomena in fermenters, essential mass balances for bioreactor design, microbial cells kinetics, fermentation simulation models, oxygen transfer and consumption, scale-up of fermenters, aeration, continuous microbial fermentations, fed-batch microbial fermentations, optimization criteria of fermentations. In the first sessions of the course, you will complete exercises using spreadsheet templates with a green-red values correction. These exercises will help you analyze microbial growth kinetics and determine the oxygen transfer rate and coefficient. In the second part of the course, you will use spreadsheet simulators to explore the operations of batch, continuous, and fed-batch fermentation modes. The spreadsheets will allow you to visualize the impact of operational variables and identify the optimal fermenter conditions. You have the flexibility to tailor this online course to your individual pace. Plan and structure the course according to your own schedule. Additionally, coordinate the interactions with your tutor through video meetings. If you would like to learn more about our online courses learning approach and teaching methodology, you can watch the video, How Our Online Courses Work. For further information on the course, please visit our Bioprocess Engineering online website or contact us at bioprocessdigital.com.